evening, everyone. Today I'll explore the relationship between farming pesticides and their effect on local Virginia soil types. Before we start, a little bit about myself. My name is Matthew Hurry and I am a senior at Gretna High School, where I'm an active member of my school community. In the fall of 2023, I plan on attending Virginia Tech to major in meteorology and atmospheric sciences. As a person coming from a long lineage of farmers, I sought a research question that would benefit both the environment and my local farming community. I chose to target the issue of pesticide contamination. By testing local soil types and compositions against a common pesticide, I can focus on the best performing soil composition for local farms. Knowing the most resistant mixture of soil could help farmers against contaminating local waterways. This subject is unique in the sense that every location is different and therefore reacts in a different way with each pesticide, meaning that my research helps add to the local database of information. As I began researching my topic, I wanted to start at the source of the issue. What is causing the use of pesticide and pesticide research to increase so rapidly? In a study by Dodd et al., he emphasized the rapid expansion of agribusiness is due to rising food demands all over the world. With millions of tons of pesticides being used each year in the EU and in the United States, chemical companies are bound to create stronger and stronger chemicals. Along with Dodd, authors Prosser and Raya agree that this race for innovation makes the jobs of researchers difficult because they must compete with these manufacturers. With this increase in pesticide production, new laws are implemented for the protection of the environment. Researchers Banks and Dos Santos state that this issue is that lawmakers must balance the needs of the environment with the needs of the lawmakers. So although these laws are implemented, they are somewhat ineffective. This led to my next topic. What makes these chemicals so bad for the environment? Studies by Dodd, Raya, Dos Santos, and Prosser all discuss that pesticides are causing enzyme alterations in organisms, and they have each conducted experiments studying the high heavy metal contents within the pesticides. Heavy metals occur in pesticides and can find their way into the diet of nearby organisms, leading to sickness and occasionally death. It has been found that the chemical composition of other pesticides can lead to enzyme malfunctions and cancers within organisms. Although it does not affect many large organisms, these changes can lead to the death of the entire colonies of microorganisms such as helpful bacteria and algae growths. My final step of research was to see how researchers classify what types of soil are most resistant to the influence of pesticides. Throughout my reading, I found that there are many different field and lab tests conducted by researchers to test the soil composition and the amount of pesticide resisted. In this literature, researchers like De Rebello, Dos Santos, and Durbig preferred to use mass spectrometry to analyze chemical makeup. Another contributing factor to an area's pesticide resistance is its slope and its soil's grain porosity. The steeper an area, the better the pesticides run off. The coarser an area, the better the pesticides seep into the soil. One major echo I continued to find in the literature was that organic matter played a heavy role in the resistance of pesticides. The higher the organic matter concentration, the more chemicals it could hold. With this literature taken into account, my study aims to fill the gap in the literature regarding how Virginia's soils specifically react to pesticides. This led me to the formation of my research question. How effectively do different characteristics of soils found across Virginia filtrate agricultural pesticides out of watersheds? From this research question, I formed my research hypothesis and my null hypothesis. My research hypothesis states that if a native Virginia soil type contains a high organic matter concentration, then that soil will capture more harmful chemicals from the soil than those without. My null hypothesis then states that if a tested Virginia soil type contains a high organic matter concentration, then it will not differ from the inorganic soils. For my independent variable, consisted of the type of soil composition that I tested. For this experiment, I used soils containing different amounts of organic matter, seeing that organic matter was directly related to the relationship between the effectiveness of a soil and its resistance to pesticide. The sandy soil contains little to no organic matter, the pamunkey soil serves as a medium, and the organic potting soil serves as a heavy concentration of organic matter. The dependent variable of my experiment focused on the pH value of the water changing, although I did collect data on other areas of chemical composition. For the control of my experiment, I misted 15 of my 45 samples with water in order to have a solid basis to compare my pesticides to. Many things were kept constant throughout my experiment. including the temperature of the testing area, the amount of time the pesticides were allowed to seep into the soil, and the amount of time each test was exposed to the sample water, and other factors such as the amount of soil, water, and pesticides applied. To begin my experiment, my father and I collected bins and wood used to make each basin for each sample. We constructed a model that contained room for 15 samples to be tested at once. We then gathered our soils from several different places to ensure that their tests had scientific validity. After this, we ordered our testing strips and pesticides. To begin conducting the experiment, I shoveled 530 cubic centimeters of respective soil into each basin. And after leveling each slot out and taking measurements, I misted on 4 ounces of permethrin tin into each basin, being sure to apply directly above the soil so that as much pesticide as possible reached it. The pesticides were allowed 24 hours to soak, and the samples were tested the next day at the same time. For my statistical analysis test, I ran a one-factor ANOVA test comparing the pHs of the 3 different soil types to one another. With a test significance of 0.05, my experiment produced a p-value of 0.01, and my null hypothesis was rejected. This confirms a diverse relationship between soil types and their interactions with these pesticides. As suggested by the chart, all recorded values were between a pH of 5 and 7, meaning that the combination of soil, water, and pesticides resulted in a more acidic mixture. The 10 test trials for Pamunkey soil and organic soil only resulted in 1 trial each where they displayed a pH other than 6, whereas the sandy soil had a wide variety of pH scores throughout the trials. This wide variety of pH tends to be higher than the other 2 soils, signaling a stronger pH neutral solution. With the aforementioned p-value of 0.01, the statistical significance shows that when testing the variables, there's a large enough difference between the groups to insist upon a further investigation into this relationship. Upon further investigation, it can be concluded that the tests within the sandy soil testing area are closer to a pH neutral balance, indicating that sandy soil harbors some sort of resistance to this acidic pesticide. The results from my experiment suggest that sandy soil is more resistant to changes in pH as opposed to other, more heavily organic soils. This conclusion is interesting as it seems to conflict with the literature that this study is based on. The strength of my experiment, however, is inconclusive due to a strong correlation within the test, but conflicting ideas with the standing literature. One factor, grain porosity, could have a strong effect on the outcome of this test. If the results of my test were to be concluded significantly on a larger scale, researchers might find different correlations between these variables. A large-scale experiment could see a slight shift of the agricultural community to more coastal areas because of the sandy soil's ability to withstand pesticide contamination. Some limitations of my study include the usage of convenient soil samples. With hundreds of different types of soil in Virginia, it was difficult to narrow down the study to types of soil that had specific amounts of organic matter within them. The largest limitation of my study, however, would have to be the use of testing strips that I ordered performed chemical tests. While they seem semi-reliable, their affordability compared to other tests could be a lurking variable. In addition, larger sample sizes could result in more identifiable trends. Although each sample consisted of 15 basins of soil, the experiment could be repeated on a much larger scale. Based on the findings of my study, further research could be done to determine if grain porosity had a data-altering effect on my experiment and others similar. Based on the results, which were contrary to the published literature, it would be interesting to see if my experiment, if it were to be completed on a larger scale, would the results remain the same. This further research could be embedded into the ever-expanding field of agricultural research in order to change the way the agricultural community views their chemical use while farming. While researching this topic, a few key themes stood out to me as a student. I learned a deep and applicable knowledge of the inner workings of a proper research project. From the literature review to keeping a detailed data log, all of the applied components of this project will serve useful in college. I am entering the field of environmental sciences, and this research helped deepen my understanding of certain aspects of the fields of natural science. To conclude my presentation, I would like to thank the judges and audience for attending tonight. Your attention and feedback is priceless. I would like to thank the PGS teachers who aided heavily in the development of my scientific process and the interpretation of my data. I would like to thank my father for being my lab assistant and for helping me to perform the experiments. And lastly, I would like to thank Ms. Long especially for her prolonged exposure to my barrage of questions regarding the APA format. And now I would like to open the floor for any questions. Yes, Ms. Long.
I was wondering where you got your soil samples. I didn't hear that.
So they were honestly collected around my hometown. You asked where I got my soil samples from. They were collected from around my house. The sandy soil samples came from a nearby creek that one of our neighbors had actually gotten sand from for a different project, and we just used the sands. That came from a local creek. The Pamunkey soil was gotten from a backyard of a farm that was nearby that had soil samples taken because they were doing, agricultural farming, and they wanted to know what best plants to grow in their soil. And then our organic soil sadly had to be bought at a store because we found that that was more convenient than taking soils and chopping up plants and then tilling it all together. So sadly, that is where the organic soil came from, but the other two were sourced from my house or around my house.
So Matthew, when you poured your soil samples into your contained basins, did you pack the soil or did you just let it naturally fall?
So Dr. Mark Jones asked if I packed my soil into my soil basins when I constructed my models and poured the soil in. The way the models were constructed is they had a door screen lining the bottom so that the pesticides and water could seep through and the dirt couldn't get through. So we did not pack the soil for fear that it would tear the screen out of the bottom of the basins.
Okay. Follow up then. So your soil's there, and then you mixed it with the, and then you poured water on top. Is that how you did that?
So the way that the experiment was conducted, Dr. Mark Jones asked if I applied my pesticides and then applied the water. So the pesticides were mixed with water in a spray bottle, and a basin of water sat below the test model. And the goal of what I was trying to, the goal of how I conducted my experimental setup was the basin of water underneath, I would spray the pesticides on top, and then whatever, 4 ounces may not seem like a lot, but when it's a basin that big, it turns out to be a lot of pesticide. So when you misted them on top, the soil became damp and heavy. So I allowed it 24 hours, and just any precipitation that misted down into the basins was then tested. Yes, Mr. Shaw.
Would you expect your findings to hold in neighboring states to Virginia?
I'm sorry, could you repeat that question?
Would you expect your findings to hold in neighboring states to Virginia?
So Mr. Shaw asked, would I expect my findings to hold in any neighboring states to Virginia? From my research, I found that the soils in Kentucky and North Carolina are very similar to those in Virginia, but the only issue with that being is my study conflicted with the literature that is based on. So until it could be repeated and a more identifiable trend that aligned with the literature could be found, I would not suggest that you would use this information in other states. Yes, Mr. Mayor.
So first, Jennifer took my question, so I've got a backup. I knew you should ask that. So since you were working with your soil samples and your water purification, was there any thought to interact with the local DCR office or Department of Environmental Quality or even the local ENS office regarding the soil and the water?
So the soil and the water, so you asked if there was any afterthought, well not afterthought, but precursor thought to check with the environmental offices and everything before I began doing my experiment. And I did not talk to any of the offices, but I did make sure that the permethrin tin that I got it falls under the umbrella of a pesticide that's actually an insecticide used for livestock animals. And on the bottle, it lists all the places that it cannot be used and all of that good stuff. And on there, we made sure to grab a pesticide that if it were to come into contact with any of the environment around my house, it would not cause any issues. Yes, Ms. Brooks.
What instrumentation did you use to check your pH?
So, Ms. Brooks asked what instrumentation I used to check my pH. Originally, I was going to use the pH testing strips from Gretna High School, but they were in very sad condition, so I had to order testing strips online. And as was listed in my limitations, the testing strips were kind of cheap compared to the other more high quality water testing kits, because I tested a lot more than just pH, because originally I was going to include more than just pH in my experiment. But I ended up going with pH just for time and, you know, ease of management. And the testing strips that I ended up buying did seem accurate, and when compared to a good testing strip that came out of the lab from the Governor School, they seemed reliable and they came out with the same pH. But like I said, their affordability compared to other tests could have been a lurking variable in the collection of my data.